In this lecture, we will be taking a look at both the botanical and entomological evidences that can be connected to the work of forensic anthropologists and forensic specialists. When studying the interaction between cadaverous material and the surrounding natural environment, one of the key areas in this study is the work of anthropoid species. Now, when you think about anthropoids, generally these are species of insects, primarily flies, but it includes beetles as well. These animals, in a sense, feed or live or breed in or on a corpse, depending on their biological preference and the state of decomposition that's associated with the remains themselves. Anthropoids, in a sense, are a really good resource for entomologists and especially forensic entomologists because they are the largest and most important group in terms of the biology of the planet, the biosphere, and of course the food webs associated with our numerous ecosystems. They can be found also in a wide variety of locations, and so they become a prevalent factor or character in the connection between, again, cadaverous material and the environment. Now, this is nothing new in terms of the work of botanists, the work of entomologists, and forensics. Botanists and entomologists have been a key player or factor in forensic research, at least in the United States, since the 20th century, at least the early 20th century. However, this study, though, can be traced all the way back to early historic China. The first documented forensic entomological kind of report writing can be connected to a series of cases, legal cases in China in the 13th century. The death investigator in this case was Sun Tzu. His medical legal contributions resulted in a variety of texts that were produced in order to take a look at, in this case, blood traces and the prevalence of blowflies and their life cycle as it related to the amount of time that the corpse was left in an exposed environment prior to being discovered. Other developments can be traced to Europe. There were very early cases of entomological research in France. We see them being, again, an important part of the investigative process in the 15th and 16th centuries. And then as we move kind of across Europe, we start to see a German school of forensic entomology and botany developing in the 18th and late 19th centuries. And that's really where we see the roots of the kind of forensic entomology and forensic botany that is occurring today, where those roots initially are developing. In the 1880s, the work of Hermann Reinhard becomes probably the foundational contribution to the development of this specialized discipline within forensic research. Dr. Hermann Reinhard, a German medical doctor, is recognized as being probably the kind of the father of this specialized approach, looking at any sort of natural evidence that relates to the decomposition of human remains and tying that in to establishing some sort of PMI determination. PMI, of course, in this case, relates to that post-mortem interval, so the amount of time that has transpired since the death event itself, and the life cycle of particular kinds of fungus material or bacteria, or in this case, the work of common arthropods, or even plant material itself and its growth and development. All of these become important resources for being able to establish that timeline and understand something about the primary or initial context within which the remains were deposited. The work of Reinhard was focused, or was inspired in this case by the systematic study of mass graves and the the remains of individuals that had fallen ill and eventually died as a result of contagious pathogens that were present in many urban environments. And basically, he was looking at, again, the contributions of insects to the decomposition process. He was also looking at flora material or plant material that was associated with decaying corpses. And all of this became important with regard to the development of not only a better understanding of decomposers and consumers at this kind of level within an ecosystem, but also the timeline that we can associate with not only their life cycle, but their contributions to the decomposition and consumption process. Fast forward to the 20th century, we see both the work of forensic botanists and entomologists taking shape in the United States or contributing to investigative studies in the United States as well. And this then becomes a specialized form of forensic study within criminology and the criminal justice environment. Your authors point out the contributions of botanists as it relates to a very early 20th century national case, or a case that received national attention, the kidnapping of the Lindbergh baby in the 1930s, as being a good resource for understanding how and why this highly specialized field becomes part of the investigative process. Again, wood debris, wood fragments from the actual ladder that was used to kidnap the child was something that was studied or analyzed very closely by botanists, and they were able to pair, in this case, the wood fibers with the actual evidence, the physical evidence of the ladder itself. So your authors kind of break these into two areas of study and looks at them a little more closely that way. I'll be doing the same thing, and we'll start out with the work of forensic botanists. The nature of botanical material, of flora, in a sense, needs to be understood in a hierarchical manner. When you think about the taxonomy of a particular ecosystem, you realize that you break things down into scaled elements. Well, macro elements and micro elements are both important to the study of time as it relates to decomposition, growth and development, whatever the case is with regard to, in this case, the criminological study. So macrobotanical elements are elements that are associated with woody stem plants, including trees and shrubs. And basically what is being studied here is their growth and development. So the seasonal layering of vascular plant tissue over time as that macro botanical element is growing and developing, that becomes important for understanding something about seasonality and, of course, the microclimate associated with a space or an area and what's happening, rainfall patterns and soil nutrients in an area and nutrient uptakes, and how that is, in a sense, varying over time. And what the botanists are looking for, in a sense, is they're looking for disruptions in the growth cycle. Again, seasonal fluctuations in important nutrients that these macrobotanical elements in that ecosystem or habitat need to survive. And you see that imprinted, in a sense, on the overall body of the material, what we're looking at in this case, which is the plant. This same kind of attention to detail as it relates to the plant tissue of macrobotanical elements can help individuals also connect physical evidence, in this case, that is outside of its traditional ecosystem. The idea of a wood fragment or a floor plank that demonstrates particular kinds of growth patterns that can be matched from one particular location to the other. Um, Trace elements in a sense. If you take a look at lots of wood products that were produced at or around the same time from a particular location, they will have similar patterns of growth embedded within that plant tissue. And that kind of tissue material, as it relates to, let's say, construction products, can be then directly associated with a kind of a primary context. If something was taken from one location, moved to another, you can match the growth patterns in that plant tissue. You can also take a look at the age of particular kinds of plant materials. You think of the specialized study of dendrochronology, where we have professionals from across a wide variety of academic and professional fields looking at the development of this vascular plant tissue over time and its overall thickness to get an understanding of the age of the material itself. All of these qualities of, in this case, plant life then on a macro scale can be documented and can contribute to the overall understanding of not only the material that might be associated with the crime scene, but also the context of the crime scene itself, maybe a primary or secondary location where the remains were being recovered or moved to. On the opposite side of this scale, we have microbotanical elements, and this is another area that is distinguished in your readings. In this case, a very common microbotanical that is used in forensic research relates to pollens. So pollinologists study pollens. They tie pollen production to particular kinds of plants, particular kinds of years, and particular kinds of in this case, climate environments. Pollen research has helped not only forensic specialists, but geologists looking at things like the Ice Age and the impact that it had on a global scale with regard to important fruit-producing and flowering plants, and how that can be again traced in terms of pollens and pollen deposits in things like ice cores and some of these other natural resources that tell us something about the changing environment. But in terms of microbotanicals and pollens, again, what is referred to as pollen rain is something that can be acquired from clothing or from the actual remains themselves. Even the lining of the respiratory system can have evidence of pollen and pollens, wind-blown pollens that were present at the time that the individual was alive and respiring, or in this case, if they were deceased, where they initially were um, deposited in terms of kind of that surrounding environment. So microbotanical elements are important in two ways, both for direct contact with plant materials, and this is where the actual pollen material would kind of leave its impression or imprint on the individual itself. Animal pollinating kinds of situations in ecosystems are a very good example of this. Brushing up against a flowering plant that is in a process of distributing pollen, or reproduction in this case, this is something that can be left as a trace element on clothing and skin and things like that. And as I said, then there can be also indirect, the idea of wind-blown kinds of pollens that can be respired. Here in the Midwest, dandelions, as we move into the kind of mid to late spring environment, these become important pollinators, and these are things that cause many people to have sinus problems and to have or experience an uptick in their allergies as it relates to particular kinds of plant species. Other kinds of grass plants also, ragweed, things like that. These are all things that will be important to understanding kind of the season at which a particular event is occurring, or what people are being exposed to, and what season it may be at the time that that exposure is occurring. And again, these can be direct or they can be indirect in nature. So creating these kind of pollen profiles for microclimate and macroclimate environments becomes very important. With regard to climate and temperature, pollens as microbotanical evidences can also be important. The idea that there may be a hyperproduction of a particular kind of plant pollen as a plant population increases in size or as it is flourishing as a result of some sort of short or long-term temperature change or change in precipitation. Because when we're looking at climate patterns and trying to tie them to kind of microclimate evidences or trace evidences, one of the things we have to consider is this kind of ratio between changes in temperature and changes in precipitation. In fact, temperature and precipitation are the key variables in identifying and defining climate with regard to the work of geologists and ecologists. So the pollens themselves then and their um, populational expression over time will be something that can be an indirect marker for climate change as it relates to temperature and precipitation. Beyond the microbotanical, we also have microorganisms and microorganismal evidence that can also help to reconstruct a death scene environment or to determine the PMI given a particular circumstance. Algae and the development of other unicellular forms, diatoms within cells. These are all things that can be very, very helpful for being able to understand the nature of an environment and even what's happening within the physiology of the remains themselves in terms of water uptake and the source of that material. Of course, algae are great for understanding that kind of larger habitat environment where we find remains, the idea of aquatic ecosystems and their condition at any time of year. So, in spring, with the warming of local environments, we see a change in the populations as they relate to things like algae. And we see algae blooms as temperatures increase. We also know that over the history of an ecosystem and a habitat, we will also see patterns of populational change as it relates to some of these unicellular or very small multicellular kinds of forms. So these are things that also provide evidence to inform botanical evidences, macro and micro, but then also giving us this understanding of a timeline as it relates to decomposition. Fungal evidence is something that has gotten a lot more attention over the past decade of research in this kind of specialized subdiscipline in forensic research. This can include the development of particular kinds of molds, molds that will be growing on natural kind of textile materials versus the remains themselves of organisms, the development of yeasts and mushrooms, toadstools, all these different kinds of characteristics tell us something about the moisture levels within an environment, the kinds of soils that are native to that environment, or the kind of soils that were native to the primary context as it relates to the death scene. There are particular kinds of flora that will thrive in cadaver situations, cadaverous kinds of fungal material that develop in association with skin and bone are great resources for being able to understand, when and where a death event occurs, and the presence of particular kinds of ferromagnetic material and ferromagnesium kinds of compounds and how prevalent they maybe are in local soils, soil horizons, and how those different horizons are being expressed in debris that's recovered from a crime scene. All of these give really good information with regard to not only how long it's been, but the circumstances surrounding a death event and its decomposition. So being able to identify what particular species of unicellular and multicellular organisms grow and thrive in these different settings has been very, very important. In this slide, I identify the Erotium genus as being one particular fungal kind of organism that we very much associate with the condition of skin, surface conditions of skin, and then even as through exposure as bone is starting to be exposed to local elements and soil conditions, there too we start to see the development of this particular genre of fungal material. Again, fitting more into that cadaverous group. Some of the ammonia-based fungi are also important to decomposition and understanding the kind of nuances having to do with human decomposition, and then those that relate to putrefaction and a lot of the gut culture of an individual, especially that associated with the small intestines and how that spreads throughout the decomposing remains in this case. As I said before, and I have or returned to this topic of pollens because they are so pervasive. The windblown pollens versus those that are the result of brushing up against a flowering plant. The Anemophilus category of plants generally are things that we find in grave soils, even historic grave soils, and these are again wind blown or wind pollinating kind of plant groups. Autopsies are something that can be used to reveal pollen deposits associated with the respiratory system. Also, the development of fungal spores inside of the lungs and the upper respiratory system, including the sinuses, the paranasal sinuses, all of these become wonderful resources. I will say, though, that generally when autopsies are being conducted, these kinds of microbotanical and microorganismal elements are not things that people specifically are looking for. However, given the circumstance of a discovery of remains, sometimes these can be brought in as important considerations and added to case reports. A lot of the specialists that will be brought in to analyze these particular kinds of faunal and floral elements and evidences generally are being drawn from academia more than anywhere else, but the understanding of what these evidences can tell us in terms of environmental issues surrounding the decomposition process are things that are, it's an awareness that we find throughout the forensic landscape here in the United States. And then, of course, as I mentioned before, when we're dealing with freshwater or marine environments and we're recovering remains from those aquatic ecosystems, this is another one of those considerations to take a look at, especially with regard to the stomach and the contents of the respiratory system. Algae and the presence of diatomic material within the remains as they are developing. And it doesn't necessarily have to be evidences directly relatable to the corpse or cadaver, but it can also be things that are in that associated landscape. So if we have a forest environment and we take a look at what kinds of decomposition processes, what kinds of consumers, ecological consumers are found within that kind of developing O horizon in a forest environment, we can reconstruct the time, the nature, and the circumstances surrounding death and a deposit. So all of this, what we've been kind of talking about in this brief lecture, relates to forensic entomology and or connects to forensic entomology in some way, shape, or form, because you cannot really isolate or look at macro or micro botanical elements and some of these unicellular and multicellular small organismal kinds of evidences without considering, in this case, some of the larger players in that decomposition process. And a lot of the early research that led to a development of this kind of specialized forensic practice really focused in on flies and their life cycle. So in the mid 19th century, larval kinds of infestations of remains, the working of maggots, and the development of adult forms of insects all become or became important considerations and characteristics to record. And one of the early species, more common species that we associate with the decomposition of human remains was the blowfly. And you've read about the blowfly in some of the associated readings that are part of this course. But the blowfly is just one part of the story. And I've got a slide coming up here in just a moment that's going to itemize a few of the other major insects that are important to understanding or establishing PMI. But it's not just the history of the insect itself that is important, but it's what the developmental stage that the insect is in when it's interacting with the corpse that can tell us even more about the PMI. Fluctuations in temperature and how that impacts populations, especially of flies, and their prevalence. Colony counts. These are all things that very much help to dial in the timing of death and how much time has occurred or how much has transpired since the death event occurred, and relating this to putrefaction and all these other kinds of processes. So the idea here is that you know the temperature, the precipitation, some of these important climate-based measures will indirectly reflect themselves on the condition of the colony of insect that is inhabiting the remains. And usually experimental studies have given us a kind of a baseline for understanding this. So here are some of the other, and this is drawn from your text, here are some of the other major players in terms of insects that will colonize a corpse, and not just human, right, but any sort of usually larger decomposing one-time living organism that now is being exposed to the elements, and they will infest it. But the blowfly is kind of the major player, but you can see there are other flies and beetles that are, and even ants that are part of this process. So look this over and relate a lot of the kind of general ideas that I'm sharing with you in this lecture to your previous readings and your continued readings this week, because this is another area that is continues to develop and is one that has been very, very fruitful with regard to being able to determine the location of a death event and the amount of time that has transpired since that death occurred.